So synthetic biology, what is it? That's our next big frontier in biology, is to take a synthetic approach, which is to say, take this approach of building new biology, and not just in the way that we've been doing it. We've been doing some genetic engineering for some time, but it's really not just genetic engineering. We didn't have the tools available to us as when genetic engineering really started. So we want to put genetic engineering, we want to add engineering back into this. And so it is, in effect, what the idea is a new interdisciplinary area of research that's going to really use engineering and biology together in a way that hasn't really been seen before. We want to have like the same sort of principles as engineers. I want to build a bridge. We want to be able to say, I want to build an organism that produces ethanol, or I want to build this cell that does this, or whatever that is. And we're getting toward that end. And we're getting toward that end by combining all of the tools that we have and all of our knowledge in these other domains and then forcing them into biology. For instance, like computer science, putting computer science into biology with the tools we have, and then engineering, chemistry, all of them, physics, all of these ideas. If we can put these together and put them into one new field called synthetic biology, we could get a lot further with our understanding of how things work and build things and do stuff for us. What this all boils down to. Let's watch a video.
After 10,000 years of genetic manipulation via selective breeding, humans finally gained direct access to the genetic code. Deoxyribonucleic acid, DNA. Since then, we've cut and pasted it, photocopied fragments of it en masse, sped-read it with sequences, printed out the code, letter by letter in the lab, modelled it on computers, and measured it with microscopes. For 40 years now, we've called this work genetic engineering. The trouble is that while there's been an extraordinary amount of genetic discovery and manipulation, there's been precious little engineering. Engineers are frustrated by genetics and molecular biology. The experiments are too slow, the complexity too messy, and growing more so all the time, and there's a frustrating lack of standardised components. They'd like to do to genetic engineering what engineers have done since the Stone Age: collect, refine, and repackage nature so that it's easier to make new and reliable things. Engineers want to treat DNA more like a programming language. Instead of ones and zeros, A's, T's, G's and C's. They want to use DNA to write simple, Lego-like functional components, inspired by but not found in nature, and then run them in a cell instead of a computer. The only difference is this software builds its own hardware. They call this re-engineered genetic engineering synthetic biology. Nowadays, rather than cut and paste the DNA sequence out of one organism and into another, you can, if you know what you're doing, just type your DNA sequence into a computer, or copy it from a database, or even select from a growing component catalogue. And then you just order it over the internet. Yes, really. The DNA sequence may be copied from nature, but the DNA itself is made by a machine. It's synthetic. The raw material for synthesizing DNA is sugar, $25 of which will buy you enough to make a copy of every human genome on the planet. The chemical letters are fed into the DNA equivalent of an industrial inkjet printer. In goes your sequence information, and out comes DNA, at a cost of less than 40 cents per base pair, and getting cheaper all the time. It's then freeze-dried and shipped to your door. Already engineers have assembled an open-source catalogue of over 5,000 standardised components, called BioBricks. At an annual worldwide do-it-yourself competition, University students build new and more complex biobricks, string them together, and then run them inside a much-studied intestinal bacteria, E. coli. Sure, they're toy projects with shoestring budgets, but the results are impressive. E. chromai. An E. coli with sensitivity tuner and colour generators is programmed to turn one of 5 colours when it detects a certain concentration of an environmental toxin. E. coloid is a bacterial system which switches on and off in response to red light and acts like a bacterial Polaroid camera. Groups with more time and a lot more money are rewriting, or call it in computer programming, refactoring, whole systems. Jay Kiesling, chemical and biological engineer, and his team at UC Berkeley have built and continually refined a new metabolic pathway in yeast by assembling 10 genes from 3 organisms in an attempt to produce synthetically the anti-malarial drug artemisinin, and to do it cheaply enough to treat up to 200 million malaria sufferers each year. Biotechnology pioneer Craig Venter has gone even further. His team has entirely replaced the DNA of one bacterium with a synthetic copy of DNA from another naturally occurring species, and added a few extras, like their email address. This wasn't creating life, it was testing just how reprogrammable a bacterial cell can be. An important step if we want biological factories which can be tasked to make many things, like vaccine, medicine, food, and even fuel.
Okay, so let's take this idea that J. Craig Venter is working on, building this minimal genome. This is a synthetic biology mainstay. We want to build an organism that's got the smallest possible genome so that we can add things to it. And once they're added to it, we know exactly what everything does. And so this was published maybe 4 or 5 years ago now. Cynthia is the name of the Cynthia is the clever. It's a synthesized genome, and it's taken 473 genes and put them into another organism. And so they've taken away every single gene individually over time, over the entire genome, and they're left with only what's absolutely fundamentally necessary. So this is really good to have as a starting point for building some sort of molecular factor. When doing this, they still can identify 149 of the gene functions. As in, there's 149 genes in there that they're like, doing something, because I can't take it out. If I take it out, I kill it. So this tiny little organism is really quick at doubling, and it's really, you can now think of this as something that we can put genes in that will make something, and then if we can make something with little else of cellular product, then we can really start to turn our biology into an engineering project. At least that's the hope here. So that's our synthetic biology example. I'd love to linger on this stuff, but we just don't have the time. We've got to go and get ourselves into some emerging infectious diseases. So we're in the middle of a pandemic, so it seems as though, like, of course this stuff is important, but and I taught this class last year at this time, it was not on anybody's radar that, hey, we could be in a global pandemic like within a couple of months. So I had gotten a couple of videos together for, actually, first I got to get some vocabulary out of the way. We need to use the word pandemic and not epidemic. It's going to drive me nuts if you use epidemic instead of pandemic. Epidemic is small little circles of major disease spread in contained areas, whereas the pandemic is everywhere. Just get that right and don't worry about it. I'll probably test on it because I get so annoyed by it. Okay, so back to this idea of, okay, we've got emerging infectious diseases going across the landscape potentially all the time is what they're going to say in this video. This is from 2018, and it was kind of ringing some alarm bells of, hey, we need to really start paying attention to this stuff because it's going to change the face of humanity. Hey, it seems to have changed the face of humanity. Let's watch the first 2 minutes of this, and then we'll get back to it.
If we look at the 20th century, the numbers of people who died from infectious diseases went way down.
The new era of technology. We know that in the cars we drive and the entertainment that we have and the way we travel and the breakthroughs in medical technology. The new antibiotics and antivirals, the cancer medicines that we see coming down the line is sensational.
We didn't have this kind of capacity back in 1918.
This year marks the centenary of the worst pandemic to strike humanity since the Black Death, the Spanish flu. It started out like any ordinary flu. People suffered from sore throat, fever, headache. Then it went on to become more sinister. It attacked mainly those in the prime of life, mainly men. In 1918, there were no antibiotics.
Doctors recommend aspirin.
Really, the only medicine that doctors had was aspirin, the so-called wonder drug.
Make you feel better fast.
It infected 1 in 3 people on Earth. It killed perhaps as many as 100 million people.
Influenza viruses are tough to deal with because they're always changing. Mutations means the existing immunity that we have in our body don't work as well and can lead to a pandemic.
Now we are seeing 3 to 5 new emerging communicable diseases with pandemic potential every year. It's not going down. The risk is going up.
So our risk is going up. We have all of these great advances in part of the way we're dealing with infectious diseases. With respect to the biology, with respect to the bacteria, we've developed all these antibiotics. With respect to the viruses, we haven't developed as many drugs, but we had this feeling that we could overcome infectious diseases. And we were pretty arrogant about it, and maybe we didn't need to be. But as we've gone through this pandemic, we are starting to see this from a different lens. But we can look at this emerging infectious disease and say, this has been coming. We've had emerging infectious diseases on the way. We've seen 3 to 5 new ones show up every year. We're seeing old ones kind of switch a little bit and become a possibility. And what is it about the way we're living that is really making this possible? Is it our demographics, as in we've got tons and tons of people, got 7 billion people on the earth now starting to inhabit some new areas? And with all of that, are we starting to interact with animals, both wild and domestic, in new ways and in not great ways? If we look at emerging infectious diseases, 2/3 of them are coming from reservoirs within animals. And so within these animals, there must be some way to switch the viruses that are in there and the bacteria to become infectious to humans. And once it's infectious to humans, then we're into a different ballgame of potential problems. Could also be some of the other contributing factors, could be the way we behave, our sexual nature, our drug use, our hygiene. As these things change, it potentially opens us up to new infectious diseases. Certainly poverty and social inequality and inequitable stances of how we live can force some populations to be more susceptible to infectious disease than others. and lurking on everything, and we'll get into this next unit, is this environmental change, this global environmental change that we're seeing with the temperature and all the associated issues with changing rain amounts, humidity, and everything that's associated with it will change these ecosystems. And maybe when we change these ecosystems, we become more susceptible to some of the infectious diseases that are out there. And we move around so much. Oh my gosh. We buy things and we're all over the universe, we're all over the world. We've got this whole system set up that is great for buying cheap things in one location, putting them in another. Go on vacation to this location. Go do business in this other location. All of this sets us up to become incredibly vulnerable to one problem spot becomes a global pandemic pretty quickly. So let's get back into this cute little puppy dogs being a reservoir for emerging infectious disease. This is called zoonotic diseases or zoonoses. These are the natural reservoir for a lot of these infectious diseases that we're starting to see in humans are actually residing in some other animal where it's been dealt with by the immune system of that animal, and it shows up in the populations of bats or camels or whatever the infectious disease we're talking about is. And so, but then it's that stage when it transfers from that organism that is the natural reservoir to the new host, where it becomes incredibly dangerous, and the immune systems aren't ready for it, and there's a failure on the part of the organism that's now infected to survive, which can be catastrophic. Of the 1,415 microbes infectious to humans, 868 of them are of zoonotic origin, as in a lot of the infectious microbes that we see are actually normal in other organisms. It's just that they make this leap, this jump to become infectious to us. And what we're seeing is a lot of the new pathogens that we see that show up are actually from animals, some animal reservoir. So how does this happen? Well, there's 5 stages that we think of as the stages of transferring from these, natural reservoir species. Take for the instance of rabies. Rabies is known to be present in some animal species. Humans don't get it from human to human. They have to have direct primary contact with the animal that has rabies, some infected animal in the landscape, scratches and bites them, however it gets into the body of the new host, the human, and there's real big bad downstream consequences of getting rabies. But that's not that problematic when it boils down to it, because there's not so many interactions we have with the individual organisms. Now we're getting into, we graduate to the next stages. We get to organisms that transfer their pathogen to humans, but then once it's transferred to humans, now human-to-human contact can actually occur. Think Ebola. We have human-to-human contact, but it's not so much human-to-human contact that we're concerned about it becoming a global pandemic, but we are concerned about the epidemics that it causes. And then as we graduate further and further up, we get to stages where it's communicable with our respiratory system specifically. Human-to-human contact is the pinnacle of this, where we think of it major step from getting the zoonotic diseases out into the population of humans. So emerging infectious diseases these days, we think a lot about viruses, because viruses are a little bit harder to deal with, right? We don't have antibiotics to just take care of them willy-nilly. They're they're more challenging by their nature, and we don't have as good of drugs to deal with them. Here's our coronavirus, the single-stranded RNA virus, positive sense. So RNA viruses are pretty dangerous because they have a really strong propensity to evolve. They have a huge mutation rate, and so they mutate quickly, they reproduce quickly, and they contact from species to species quickly. All of these things combine to make for a very effective evolution of a virus to adapt to the new environment that it finds itself in, the human body, whoever's human body it winds up in. And so if that evolution, it's really dangerous as well, because as it evolves, it'll find a new way to survive, and that new way to survive will have potentially downstream bad consequences for people who are infected. So, okay, so that's the general idea of emerging diseases. What about just like the common flu? The flu that we see every year, we see it named the H number, N number. What does that mean? What are the H1N1? What does that mean? Well, the flu virus only has 8 genes in it. It's a tiny little virus. It's got like almost nothing there, but it kills 40 to 50,000 people in the U.S. each year. What is it doing? How is it making a living? How's it getting through our defenses is what this boils down to. So the way it gets through is it has subtle changes or it has big shifts. And so we'll get into that in the next slide, but just to take away here, we've got H1N1 would be the name of one of the viruses you might see, the flu viruses you might see. The H is referring to the receptors on the surface of the cell that interact with the cells it wants to infect, and then transports it into the cell so that it can reproduce and make a living. The neuraminidase, tough word to say, neuraminidase, okay, fine, I'll stop trying. It's expressed on the surface of the cell so it can exploit the viral particles from the nucleus. And so this combination of these two is what shifts generally over time. We have small shifts. Hey, this is just RNA that's being modified. It doesn't have any of the machinery to be modified to edit any mistakes it makes. So we're going to have genetic drift over time. So these things will just drift apart from where they were, just as a matter of course, just as a matter of typical evolution. And so we have yearly flu viruses that infect our population. It's really dangerous when we get big pull-on shifts of the virus. And so we'll get into this antigenic shift and antigenic drift here in this video. Let's go.
Pandemics, or global epidemics of influenza, have occurred for hundreds of years. There were 3 pandemics during the 20th century. They occurred in 1918, 1957, and 1968. The 1918 pandemic took the largest toll, causing an estimated 50 million deaths worldwide. The first pandemic of the 21st century began in 2009. But how does a pandemic occur? Influenza A viruses vary slightly from year to year, a phenomenon called antigenic drift.
So this drift is that subtle change over time. Think genetic drift applied to the flu virus.
These small variations are the reason why the vaccine must be updated every flu season. Influenza A viruses also can undergo dramatic genetic changes called antigenic shift that may cause a new influenza strain to which we have little to no immunity. If this strain spreads easily among people, it may cause a pandemic. The immune system targets 2 proteins on the surface of influenza A viruses, hemagglutinin and neuraminidase. Hemagglutinin has 16 confirmed subtypes of proteins. Neuraminidase has 9. From these protein combinations, scientists name influenza viruses. For example, An H5N1 virus contains hemagglutinin subtype 5 and neuraminidase subtype 1.
Okay, so we have these two ideas: the antigenic drift, that's the year-to-year change that may occur, pardon me, in the population of flu virus, and then we can have this antigen shift where everything gets shifted around. We do not have time to watch it, so we won't watch it. And you're not responsible for that last slide. So this antigenic shift is, in effect, what happens is we have a viral particle of flu enter a cell of one organism, but it's come from two different flu infections at the same time. They get together in the cell, rearrange the RNA, that becomes a new cell. That new cell is exported, and it suddenly becomes a new looking cell, new looking virus. Our immune systems aren't capable of quickly adjusting to this new virus, and so we won't be able to just use the immune system we have to effectively knock it off easily. Take its toll on humanity when we have these antigenic shifts. All of the pandemics we see are antigenic shifts. Hey, here's the Spanish flu from 1918. Just notice how similar this is, as well as the changes that have occurred. I know we're running out of time. Okay.
An estimated 20 to 50 million people died from the Spanish flu in 1918 and 1919, which is more than the estimated 17 million deaths during World War I. It's called the Spanish flu because Spain was neutral during World War I, and none of the other European countries wanted to project weakness while the fighting continued. This particular strain was so powerful that it would turn your skin blue and fill your lungs with fluid, eventually causing suffocation in sometimes less than 24 hours. World War I actually helped the flu spread rapidly, as traveling soldiers spread the virus unknowingly. This combined with the idea that all the supplies and healthcare workers were on the battlefront. The home front was not ready for the flu. In the USA, an estimated 675,000 people died, which is more deaths than any single war in American history. Some Chicago factories got so worried about the flu spreading that they put wiring around water fountains to make sure workers didn't put their mouth on them and spread the virus. Some cities were so scared they shut down all public places and quarantined all infected individuals. Morgues in the United States were overcrowded with bodies, and many people had to dig their own family members' graves. Many famous people were infected by the flu and survived, including Walt Disney, sitting President Woodrow Wilson, and future President Franklin D. Roosevelt. Many historians call the Spanish flu the forgotten pandemic because it was overshadowed by the horrors of World War I. In a highly controversial lab experiment in 2014, scientists at University of Wisconsin created a virus that is only 3% different than the original Spanish flu. Do you think scientists should be doing stuff like this?
Okay, so there's our Spanish flu. I don't know about you, but that looks really familiar. People in mass in the 1918, 1919 time period. People shutting down schools, morgues being overrun. This is a different time. President coming down with it. Okay. I hate when students would do that in school. They should bring back the. Oh, yeah. Yeah, because the mouth all touches its gut. Okay. So that's our viral section in emerging infectious disease. Turns out we have this feeling, and it's kind of well in, but really shouldn't be there, that we can solve bacterial problems. And we have this feeling because we've done it really successfully. We developed antibiotics over 100 years ago. And we think of bacterial infections as, we got this, no big deal. And for a long time, we relied on this as being a no-brainer of, okay, bacterial infection, take this antibiotic, give me in a couple days. Problem. Turns out we're doing that not just with people, but we're also doing that with our livestock. We're doing that just to keep our livestock industry growing. And we're doing it with all sorts of antibiotics. And when we do this, we're somehow reducing the overall impact of the antibiotic. And wait, how could that possibly be? Well, the way that is kind of shaping out is that we're selecting effectively for the resistant bacteria. Those resistant bacteria are spreading their resistance that they've built through the shedding of their plastic, the conjugation of the bacteria. And so we're effectively selecting for superbugs. And so things like MRSA. Let's watch a short video. Kurt Zagat's super.
Bacteria are among the oldest living things on this planet. The smallest thing we still consider life, they are masters of survival and can be found everywhere. Most bacteria are harmless to us. Your body hosts trillions of them and they help you to survive. But others can invade your body, spread quickly and kill you. Millions of people used to die as a result of bacterial infections until we developed a superweapon, antibiotics. Together with vaccinations, antibiotics revolutionized medicine and saved millions of lives. Antibiotics kill the vast majority of susceptible bacteria fairly quickly, leaving only a small group of survivors that our immune system then deals with easily. How do antibiotics do this? Imagine a bacterium as a very complex machine with thousands of complex processes going on that keep it alive and active. Antibiotics disrupt this complex machinery, for example by interfering with its metabolism, slowing down their growth significantly so they are less of a threat. Other antibiotics attack DNA and prevent it from being replicated, which stops bacteria from multiplying, ultimately killing them. Or by simply ripping the outer layer of the bacteria to shreds so that their insides spill out and they die quickly. All of this without bothering body cells. But now, evolution is making things more complicated. By pure random chance, a small minority of the bacteria invading your body might have evolved a way to protect themselves. For example, by intercepting the antibiotics and changing the molecule so it becomes harmless. Or by investing energy in pumps that eject the antibiotics before they can do damage. A few immune bacteria are not that big a deal because the immune system can take care of them. But if they escape, they might spread their immunity. How can bacteria spread immunity? First of all, bacteria have two kinds of DNA, the chromosome and small free-floating parts called plasmids. They can hug each other and exchange those plasmids to exchange useful abilities. This way, immunity can be spread quickly through a population. Or in a process called transformation, bacteria can harvest dead bacteria and collect DNA pieces. This even works between different bacteria species and can lead to superbugs, bacteria that are immune to multiple antibiotics.
So just note there that if you'll note specifically, they said it can actually go from one species to another species in this bacteria realm, which makes it even more scary because we can turn on some genes in other bacteria that could do other damage.
A variety of superbugs already exist in the world. Especially hospitals are the perfect breeding grounds for them. Humans have short memories. The horrors of the pre-antibiotic era have been forgotten. Today, we treat this powerful medicine as a commodity instead of as the game-changing achievement of science that it is. This has led to a strange disconnect. Hundreds of millions of people still don't have access to antibiotics in developing countries, while in other parts of the world, antibiotics are prescribed too freely and taken without care. Antibiotics should be a last resort drug, not something you take because your cold is annoying. Another serious problem is antibiotic use in meat production. At any particular point in time, humanity holds between 20 and 30 billion animals as livestock. To make meat cheaper, many animals are held in horrible conditions, in very tight spaces, and in unhygienic conditions. The perfect breeding ground for a disease. So many animals are given antibiotics to kill as many bacteria as possible and prevent them from getting sick, because a cheeseburger has to cost $1. Unsurprisingly, as a result of this system, we have created more and more bacteria that are resistant to antibiotics. To counteract this, we use different antibiotics, and we have another secret weapon. There are specific antibiotics that are used to wipe out bacteria that have developed resistance. There are strict rules for using these to avoid the creation of a superbacterium. Or so we thought. In late 2015, scary news arrived from China. Resistance against colistin, a last-ditch antibiotic, had been discovered. Colistin is an old drug and was rarely used because it can damage the liver. So there was little resistance against it, which made it a great antibiotic of last resort for certain complex infections that occur in hospitals to fight bacteria that have become immune to a whole bunch of other drugs. Bacteria resistance to colistin is very, very bad news. It might destroy a last line of defense and lead to a whole lot of dead people. How could this happen? Millions of animals in Chinese pig farms have been given colistin for years. Resistant bacteria developed, spreading first from animal to animal and then to humans without being noticed. On an average day, there are over 100,000 flights on Earth, kind of connecting every human on the planet. By creating the modern world, we have also built the infrastructure for a dangerous pandemic. Still, we don't need to panic just yet. Bacteria evolve, humans do research. New antibiotics are developed as old ones become obsolete. Technology is advancing every day. The problem is real and serious, but the fight is far from over. If humanity plays its cards right, superbugs might turn out to be not very soup after all. This video was made possible by viewer support.
Okay, so that's our antibiotic resistance. If you were presenting today, get ready because I'm going to say, turn it over to you in like 2 minutes. Okay, so antibiotic resistance is effectively this this kind of backwards way of looking at evolution. We effectively are wiping out populations. Only a couple survive with the advantageous genes to survive. Those genes are passed to offspring, and it shifts the population towards survival. And so that's what biology is wont to do. It wants to survive. For instance, MRSA is probably the most famous of the superbugs. It's methicillin resistant staph infection. It developed this resistance to penicillin in the 50s. It developed resistance in the 60s to methicillin. And so it's resistant to almost all antibiotics. And it's one of these things that shows up in hospitals and it's pernicious. It gets in there and it'll kill a lot of people. 2 million people per year infected in hospitals, some 100,000 deaths. It's a problem. And if we just look at the rate of antibiotic resistance development, effectively what happens is we develop an antibiotic. There goes here comes some resistance to it, and so we have organisms that can continue to develop. Antibiotics developed, resistance made. Antibiotics developed, the bacteria develop a resistance. And so all of these things are kind of coming together as this, as we're developing this like arms race. This is almost like coevolution. We're co-evolving with our microbe. I won't call them friends, our microbe foes. Okay, we have an exam on Tuesday. The exam will look a lot like our previous exams and quizzes. Your homework is to study for that. You obviously have an in class to give me any multiple choice question. Give me a multiple choice question from today's material and any feedback you want to give me. Love to get feedback. It's just makes my day when I get some feedback, especially when it's like, hey, you should do this better.